After glycolysis is complete, we have two pyruvate molecules. Now, what happens next depends on whether or not there is oxygen. If O2 is present, O2 present, we enter aerobic respiration. That means that it's going to enter the mitochondria. Mitochondrion. If there is no O2 present, on the other hand, we have to kind of double down on glycolysis. If there's no oxygen present, glycolysis is the only means by which we have to make ATP from the glucose that's available. Maybe we have more glucose molecules that we can keep breaking down and breaking down and breaking down in order to get more and more ATP, but the only way we can do it is through glycolysis, getting these two net ATP per glucose that we get. But in order to do that, we need something else. This NAD here, it's produced by taking a coenzyme called NAD+ and reducing it to NADH. If we don't have any NAD+ laying around, we can't do glycolysis. And if you're an anaerobic organism, so you don't have the option to send your pyruvate molecules over to the mitochondrion to keep getting oxidized and broken down further, well, you need to keep doing glycolysis. So that means you're going to need more NAD+. Now, you might think, okay, so it's going to go through a biochemical pathway or something like that. It's just going to manufacture more of this NAD+ from starting materials. But there's an easier way to do it. See, we already have all this NADH lying around from glycolysis, 2 of them for every time we break a glucose in half. Now, what we can do is remove that hydrogen from the end, and then that will bring it back to being NAD+. So, how do we do that? We use NADH to reduce pyruvate. We attack this pyruvate with it, and we call that fermentation. This is an anaerobic process, no oxygen. Okay. If you can get the NAD back, you can get more of this NAD+ lying around, then you can keep doing more and more glycolysis, keep breaking glucose in half in order to get the 2 net ATP. Now, why can't we just use this NADH for getting energy? Well, we can if there's oxygen present. But if there is no oxygen present, then the NADH is basically useless to the cell. So instead, we're going to use it for fermentation. And fermentation can have a number of interesting products. These 2 pyruvates, once reduced, in some organisms, you get ethyl alcohol. ETOH is a normal abbreviation for that. In other organisms, like us, you get lactic acid. Which is why, if you're lifting weights, then after a while your muscles start to get sore. You see, my muscles need oxygen in order to produce ATP from the blood sugar that I have. If I'm trying to produce way too much, I'm putting too much strain on it, and there's not enough oxygen getting to my muscles in order to give me the ATP that I need, my muscles will be forced into an anaerobic respiratory process. And in that situation, they will begin to ferment that pyruvate. They will begin to undergo fermentation and reuse the NADH and start producing lactic acid, building up inside my muscles as a kind of waste product. And this is why my muscles will feel sore after a while of doing this kind of exercise, which I'm very unused to doing. So I'm going to put this down now. So on this slide, you can actually see the kind of choice that's being made here. If there is no oxygen present, it's going to go and do fermentation. It's going to recharge the NAD by reducing these pyruvates, and then you get your lactic acid or your ethyl alcohol, depending on what kind of organism you happen to be. Now, I know that all the discussion of the coenzymes, they're just throwing a bunch of letters, right? NADH and ATP and acetyl-CoA and all these. The language gets inherently confusing here. So I like this diagram because, to me, this makes a lot more sense. It shows you glucose coming into the cell, and it's being snapped in half through glycolysis. Glycolysis is about breaking sugar. So we break it into two pyruvate molecules. And in the process of doing that, we get our ATP and our NADH, but we need NAD plus in order to do glycolysis. And you can see that as a reactant down here on the diagram. However, if there's no oxygen present, then we are going to have to recharge that coenzyme NAD. So you can see in this slide here, there's a little circle, a cycle, where we are using NAD to oxidize glucose to break it apart during glycolysis, and then we are taking that NADH that we've made and we are reducing the pyruvate in turn in order to get these different chemical products. So if you take a look down here at this chart, it shows you glucose being broken into pyruvate. That pyruvate is then broken down into an even smaller molecule, acetaldehyde. Now, we use the NADH to reduce that product, and then you get ethyl alcohol or lactic acid, depending on what kind of organism you are. And the NADH becomes NAD again, and NAD is something that we need for glycolysis. Go back to the video where I was talking about glycolysis, and there should be a little part of the diagram where NAD becomes NADH. So that's the purpose of fermentation.
Now, when you hear the phrase fermentation, it's natural to think about beer and wine and the alcohol-making process. Alcohol, ethyl alcohol, is one of the byproducts of fermentation. So it's the same process that we're talking about here. That process is accomplished by a single-celled fungi, which we call yeast. Saccharomyces cerevisiae is the particular strain of yeast that brewers use in large breweries. It's also the strain of yeast that bakers use when they're making bread. And the reason that bakers find this to be a valuable microorganism is because the other byproduct of fermentation is CO2, carbon dioxide. So if I add some of this dry, inactivated yeast along with some sugar, sucrose in this case, and then I activate them, give them a nice aquatic environment to wake them back up. We should be able to see the products of fermentation beginning to form. Since carbon dioxide is a gas and it's in a liquid environment, what we're going to see is bubbles being created over the course of this chemical reaction. Those bubbles are going to create a nice frothy foam. So I'll be back in a moment to see how this is done.
process of fermentation continued even after I mixed the yeast-water-sugar mixture into the dough. And because the CO2 gas kept building and building and building, the dough rose, the dough expanded, and we got all of these nice pockets of gas, which you can see on the inside. The structure inside of bread comes from that byproduct of fermentation, the CO2 being produced. So you can actually see the nice open structure inside this bread that came as a result. Unfortunately, yeast get kind of short shrift in introductory biology courses. We only really mention them early on at the beginning, but next time you're having yourself a sandwich or a nice loaf of bread, thank Saccharomyces cerevisiae for doing all the work of fermentation for you.